Hello guys, my name is Kirtan and welcome to my channel Physiology at Once. Today I will be talking about the topic nerve injury. This can be asked in the board exam for at least 5 marks. So this is an important topic to study. So first of all, what is nerve injury? It is an injury to the nerve tissue. The nerve tissue may include the nerve and the myelin sheath by which it is covered. So when a nerve is cut, the distal part of the nerve is separated from the cell body and it degenerates. This is also called as Wallerian degeneration. So you can understand this by, this is a nerve cell, and this is the cell body of the nerve, and this is the axon, and this is the terminals of the axon, and it is covered by myelin sheath. So when the nerve is cut here, suppose the distal portion degenerates and this portion survives and may again regenerate if the condition persists. So this degeneration is called Wallerian degeneration. So the nerve injury can occur due to many causes like a cut or a mild pressure or many more such things. So how do you grade the nerve injury? So there are many methods to grade the nerve injury. This method is based on the severity, and this was given by Sunderland. So according to this classification, the grading of nerve injury can be divided into 5 degrees. The first degree of injury, in it there is temporary loss of the function of the nerve due to the mild pressure on the nerve. The second degree injury contains severe damage with intact endoneural tube. Endoneural tube is nothing but a covering outside the myelin sheath of the nerve. Third degree injury, in it there is several damage to the nerve fiber including the endoneural tube. In the fourth degree injury, there is severe damage to the nerve which is associated with the disorganized nerve fasciculi. Nerve fasciculi is nothing but the nerve fibers, nerve fiber bundles. The fifth degree injury, it is the complete transaction of the nerve, and the regeneration is almost impossible in this injury. So what and all happens after the nerve injury takes place? First of all, there is degenerative changes. The nerve degenerates. The degenerative changes occurring in the distal segment of the axon are called Wallerian degeneration, as I talked now. The main changes are, it affects the transmission of impulse. So normally the impulse is transported from nerve to nerve, and in the last the axon terminal carries it to the next nerve. So due to the cut of the nerve or the nerve injury, the synaptic transmission does not occur at the axon terminal. It almost stops within hours immediately after the injury to the nerve. The axonal changes. What happens to the axon? It swells and the terminals retract from the postsynaptic target. Postsynaptic target can be a muscle, a bone, or anything else. It also degenerates over a period of several weeks. It also degenerates. And there is formation of round fatty enlargements of myelin sheath that looks like a series of beads. There is round fatty enlargements seen like, see, if this is a cell body and this is a nerve, here it's cut, so it makes a bead like formation. The myelin sheath makes a bead like formation. See, this is a bead like formation. This one is the bead like formation. So this word is important, a bead like formation. What is the effect of stimulation? Normally the stimulation occurs and it conducts the action potential through the axon terminal. But after the nerve injury, it can conduct the action potential only for 3 days, but after the 5th day it cannot. The changes in soma and stump. Stump is nothing but the distal portion of the soma or cell body. This is called the retrogate degeneration because it appears in the proximal part of the nerve. So what happens in this? The soma or the cell body swells and becomes rounded. The nucleus also swells and moves to an eccentric position. Eccentric position means that it moves to a corner position. The endoplasmic reticulum comes around the periphery of the cell body. It also moves to the periphery in the cell. The ribosome appears disorganized. The nissel granules, these are the granules present in the cell body, gradually disintegrate and are stained weakly with the basic dyes. This process is known as chromatolysis. So let's understand this degenerative changes through diagram. See, this is a normal nerve cell and there is a cut here. This is site of nerve injury. So the distal portion now starts to degenerate. There is also changes in the nucleus cell and the soma. The nucleus swells. The soma also swells. The soma is nothing but the cell body. The axon also swells as talked now. And there is formation of bead like through the myelin sheath. I talk now. And there is also retraction of the axon terminals. There is chromatolysis or the disintegration of the nissel granules present in the cell body of the nerve. There is also formation of axonal sprouts. These axonal sprouts can give rise to growth cone. This actually occurs in the regenerative process. If the nerve cell is able to regenerate, only then. So let's talk about the regenerative process or how it can regenerate even if it's degenerated. So in the regenerative changes, the axon develops many sprouts or growth sprouts. Any of this growth sprout can give rise to a growth cone, which finds its way through the Schwann cells and re-innervates the original target. Any Schwann cells which are degenerated gives way to this growth cone and can again reform to supply this and form the myelin sheath. The somatic changes. Number of the nissel granules reappears. The cell also loses excess fluid and regains the normal size. It will be enlarged in the degenerative changes. There is also reversible of the chromatolysis process. It can happen only if the conditions are appropriate and the damage is not so severe. What are the changes in the target structure? Target structure can be a muscle or a bone or any organ or anything else. The regulators in it are upregulated. So why are the regulators upregulated? So when the nerve is injured, the axon terminals loses many nerve receptors which are important for the synaptic transmission. I will talk about the synaptic transmission in the next video. So when these neurotransmitters are damaged, there is no conduction of nerve impulse from this to the target structure. So in this response, the target structure develops the regulators and those are upregulated. So this is a feedback mechanism like type. So this is also called as denervation hypersensitivity. So because of the denervation, there is hypersensitivity due to the regulators are upregulated. Let's understand this regenerative changes through the diagram. So after the formation of the axonal sprouts, the growth cone comes and it finds a way through the Schwann cells and tries to reach the postsynaptic target. So the Schwann cells make way and they form the myelin sheath around this growth cone. So slowly the nissel granules also start to appear. You can see here. So after this, the nucleus comes to the normal size and also there is an appearance of nissel body, the reverse chromatolysis. Also the cell size is decreased due to the excess fluid loss. The myelin sheath is also formed due to the Schwann cells. The postsynaptic target also contains many receptors which occurs due to the denervation hypersensitivity. So I hope you understood this video perfectly, and please like, share and comment if you found this video useful, and also subscribe to support my mission to make physiology simple, easy and understandable. Thank you.